Hello everyone, I will now be going through the topical exercise 3.2, part 2 of energetics, now photosynthesis. As mentioned in our previous video, if you have not done so, please do a first round of self-evaluation at the very bottom. You can scroll to the bottom and look at the success criteria. Each success criteria is tagged to a particular question. For example, unable to identify what is needed for photosynthesis to occur. To see if you understand, you can do questions 5, 1A part, question 1A and B, 2A and B. Alright, so if you've not done so, please do your self-review immediately after doing the paper. Then, after listening to this video, you review yourself a second time to see if there's any improvement in your understanding. Let's begin first with question 1. Question 1 asks, which combination correctly describes the light-dependent reactions of photosynthesis? Remember that the whole point of these two topics, cellular respiration and photosynthesis, we want to ground ourselves in interactions. And in interactions, we want to know what is used and what is produced and where it takes place. And so you see a lot of our questions stem around these 3 points. So where does the light-dependent reaction take place? It takes place at the granum. Granum are, several stacks of these thylakoids. What is produced? We know that ATP is produced, so these two options are possible. Okay, ATP is not used as in option A and B, but it's produced. We also know that NADPH is produced. And if you see a lot of this in the subsequent parts of this paper, you'll see that sometimes it's described as reduced NADP. Okay, reduced NADP is the same as NADPH. So NADP is reduced, yes, NADPH is also produced as a result of this process. So the best answer is D. Let's look at question 2. The diagram shows the path taken by electrons and the formation of hydrogen ions in the light-dependent stages of photosynthesis. What are the electrons and hydrogen ions used to produce? Again, you see interactions. We want to know what's produced, what is used, where it takes place. So, as mentioned earlier, ATP is produced from ADP. And we also get NADPH, which is what this means. When they say reduced NADP, they're referring to NADPH. So, best answer is B. The reason why A is wrong is when they say NADP is produced from reduced NADPH, they are saying that NADPH is converted to NADP. But that's not true. It is the reverse that's happening. NADP becomes NADPH because NADP serves as the final electron acceptor. Okay, C and D confirm wrong because these are things referred to in the Kelvin cycle. Okay, not at all. Kelvin cycle is also the light independent stage. Alright, so A and B is either A or B, but B is the best answer. Let's look at question 3. Question 3 talks about this particular chemical, which is a weak killer. And it acts as a weak killer by blocking the flow of electrons from the electron transport chain in the light-dependent reaction of photosynthesis. And they ask, why does this kill the plant? First and foremost, A says active transport of minerals is prevented. This is not correct. Okay, I think this part is referring to what's being absorbed at the roots. So this weak killer does not affect what's happening at the roots. B, ATP and reduced NADP, or in other words NADPH, is not produced. This is true. And why is this devastating? Because without ATP and NADPH, the Kelvin cycle or the light independent stage will not be able to happen. If that doesn't happen, we will not produce any sugar or food for the plant, and the plant will eventually die. C, photoactivation of the chlorophyll cannot occur. This can still occur. Alright, electrons can still be excited, but the only issue is that they won't be able to transport down the chain. D, photolysis of water does not occur. Okay, this is a possibility. It could be that photolysis cannot occur because electrons cannot flow anymore, right? But this isn't the main reason why it kills the plant. The main reason why it kills the plant is actually part B. Now let's move to question 4. Which is an electron donor to the electron transport chain in a light-dependent reaction? Okay, I think this one is rather straightforward. The electron donor is water. Through photolysis, water will release electrons, and also oxygen will be a byproduct. And this electron will be channeled into the electron transport chain. If I flip the question and ask which one is the final electron acceptor, the answer would be, oh, actually there isn't an answer here. The best answer would actually be NADP. In the case of cellular respiration, the final electron acceptor will be oxygen. But in the case of the light-dependent stage, NADP is the final electron acceptor. Let's go to question 5. Question 5 seems challenging, but actually they are essentially asking you where you'd find the products of photosynthesis for each process. Okay, so they provided you water and carbon dioxide that is radioactive. Why are we providing radioactive isotopes, or rather water and carbon dioxide that contains radioactive isotopes? It's so that we can track where the water and carbon dioxide is going to be used during photosynthesis. That means to say, any water containing isotope of oxygen 18, you'll be able to track where this oxygen goes through the process of photosynthesis. Okay, so maybe let's just work with one thing first. Where is water used in photosynthesis? It is used in photolysis. And where is photolysis taking place? It's taking place at the thylakoid, because this is where it's going to be used as the electron donor for the electron transport chain in the light-dependent stage of photosynthesis. Where is carbon dioxide used for photosynthesis? Okay, carbon dioxide is used for the Kelvin cycle. Kelvin cycle takes place in the stroma. What's the purpose of Kelvin cycle? Is to produce the sugars that the plants require. Okay, so with that in mind, this is how we answer the question. Where is the chloroplast? Where in the chloroplast would the products of photosynthesis containing these isotopes be formed? So, if water is used in photolysis, you expect that the products that are formed from water being photolysed, okay, and the product will be oxygen. This oxygen will also contain isotope 18. So you expect that the product to be found in the thylakoid. What is this product? Specifically, it will be oxygen. Okay, the oxygen that's produced from photolysis can be found in the thylakoid. What about carbon dioxide containing oxygen 17 and carbon 13? You'll find the products, which is specifically the sugars. Okay, these sugars will be found in the stroma. Okay, so the best answer will be C. The sugars will contain oxygen 17 as well as carbon 13. Alright, so let's go to question 6. Oh, there's no question 6. Let's look at structured question 1A. State the importance of photosynthetic pigments in sustaining life. The key word you want to look up for is that absorb light energy. And what is this light energy going to be used for? It's used to produce, you can say food or starch, or you can say oxygen is produced. But this oxygen, is going to be used for other living organisms. Okay, so you could mention any of these points. Let's look at the next part. Okay, maybe let's put some context to this question. This question provides you a table of various photosynthetic pigments. Each one of these photosynthetic pigments has a maximum absorption spectrum. Meaning to say, it is at this range of light spectrum that it absorbs best. So the next part of the question asks, or rather provides you a graph showing the absorption spectrum of the chlorophyll or the pigments that can be found in light harvesting complex 2 and light harvesting complex 1. Okay, the question is saying a purple bacteria contains two kinds of light harvesting complex. Light harvesting complex 2 and light harvesting complex 1. Inside these light harvesting complex, you can find photosynthetic pigment. And what this graph is showing what this photosynthetic pigment, what's their best light absorption range. So the question first asks, suggest and explain where you expect to find the light harvesting complex in the purple bacteria. The answer is the cell membrane. Why the cell membrane? Well, because bacteria are prokaryotes. Prokaryotes, as we know it, do not have organelles. If they do not have organelles, means they won't have things like mitochondria. So, in order for the light harvesting complex and electron transport chain to work, they need to be arranged, kind of in sequence. They need to be side by side each other. The only way is for it to be bound to a membrane. And the only membrane of a bacteria is the cell membrane. So you find the light harvesting complex embedded on the cell membrane of the bacteria itself. It cannot be free floating. If it's free floating, there won't be a sequence in which the electrons flow. Okay, so this is the answer. Next, let's look at C. Identify one photosynthetic pigment most likely to be found in bacteria based on the absorption spectra. So they're asking you, what is the photosynthetic pigment that's likely to be found in LH2 or LH1 based on what you see in the graph? Based on what I see in the graph, it seems like light is best absorbed in this range, in the 800 plus range. So let's look at which pigment absorbs light best in the 800 range. And we find that it's bacterial chlorophyll A that absorbs best in the 800 plus range. Therefore, the answer is bacterial chlorophyll A. Suggest and explain an advantage of synthesizing photosynthetic pigment that are different from chlorophylls. In other words, we are asking, what's the advantage of having pigments that absorb light that is out of the range of what chlorophyll, the green chlorophyll, absorbs? Well, if it absorbs light from a range that is not what chlorophyll absorbs, then we reduce the competition for absorption of light. If both the bacteria and plants absorb the same wavelength of light, they'll be competing for the same light source. Okay? Another reason you could give is that perhaps these bacteria are absorbing light that can actually reach the deep waters of the ocean. Perhaps the wavelengths of light that are typically absorbed by chlorophyll cannot reach the deep waters. And so perhaps the bacteria is adapted to this by having pigment that can absorb light that can reach deeper waters. And perhaps the light that can reach deeper waters are the light that falls within the 800 plus range. Okay? Oh, if I'm not clear, so maybe I say it again. So perhaps this is water, right? And then we have light penetrating. But perhaps the light that is able to penetrate deeper into the water that reaches the purple bacteria, perhaps this light falls in the 800 plus range. Whereas light that plants require, or the light that chlorophyll can absorb, can only penetrate up to a certain depth of water. Okay, and this is probably the light range they found over here. Let's go to the next question. State the role of proteins P, Q, and R. Their role is that they are electron carriers found in the electron transport chain. Help to transport the electrons, yeah? F. Explain the impact of photosynthesis if Q is inactivated. So where's Q? Q is this one. So what if this is inactivated? Once again, every time you encounter a question that asks you what will happen if a particular protein is disrupted or inactivated? Actually, what we are really asking is, how does the electron transport chain work? Just that we are asking it in a roundabout way. Okay, so how will we describe and answer this question? Number one, if this protein Q is disrupted, you'll find that electrons cannot be transported to photosystem 1 to replenish the lost electrons. Right? Remember, electrons that were excited in photosystem 1 will be transported down the chain, eventually to NADP, to form NADPH. Right? And as a result of photosystem 1 losing an electron, it needs to be replenished. And it's replenished by electrons coming from photosystem 2. Okay, that would replace whatever is lost in photosystem 1. But if we knock out Q, this electron can no longer be transported to replenish this lost electron. Point 2, as a result, NADPH cannot be formed, as I was saying, due to the lack of electrons for reducing NADP. The electron transport chain will fail to function, and as a result, the proton gradient will no longer be set up. You see the usual thing. It's very similar to questions related to cellular respiration. If the proton gradient is no longer set up, you'll find that ATP synthesis will be reduced because there will be no flow of protons across the membrane. And if we have a lack of ATP and NADPH, okay, if these two things are no longer produced, it will also affect the light independent stage, which is where the Kelvin cycle takes place. The Kelvin cycle can no longer function as well because you lack NADPH and ATP. Okay, so this is a rather long answer. Any one of these 4 lines will do for you. Next, let's go to the next question, question 2. Now, question 2 is not something explicitly taught in class. Okay, so the answers you're about to see will be a little bit foreign to you. Take it as learning for yourself. It's not out of this world, but it's not something we taught in class either. So with reference to the structure you see, explain how the structure is related to the function in photosynthesis. Number 1, thin cell wall and cell membrane. I would like you to not look at the cell membrane part. You cannot have a thin or thick cell membrane, but you can have a thin cell wall. Thin cell wall means that cell wall has less cellulose. Thick cell wall means there's a lot of cellulose there. So what's so good about a thin cell wall, as you can see in this diagram? Well, if you have a thin cell wall, it allows essential nutrients or raw materials like water, CO2 to reach the chloroplast, which is inside the cell, much faster and easily. Next, you'll find that there are a lot of chloroplasts present in this plant cell. Numerous chloroplasts. There's a structure. And what's the function of it? To maximize the absorption of light energy. Last but not least, you see that there's a large central vacuole in the plant cell. There's a structure. What's its function? Is to store photosynthetic products like sugars. So I would say this point in the middle perhaps is something you could have written. These two may be a little bit out of this the syllabus, but it's hopefully something that you may have inferred or suggested. Let's go to the next question. This question here shows an electron micrograph of a part of the chloroplast showing thylakoid membranes. Okay, so actually if we were to zoom in, this is the outer membrane, and then this is the inner membrane, and then these are the thylakoid membranes of the thylakoid. Describe the role of the thylakoid membrane in photosynthesis. Now this seems like a very difficult question to answer, but actually we're just asking you things related to the electron transport chain. How is the thylakoid membrane relevant to the electron transport chain? Okay, number 1, the thylakoid membrane, right, has a large surface area for light-dependent reactions to occur. Because all the thylakoid membranes are folded to form these small little thylakoids that stacks up into grana, you increase the surface area for all the electron transport chain proteins to be embedded. Next, how else is the thylakoid membrane important for photosynthesis? The thylakoid membrane is where you can find a lot of photosystems embedded to trap light energy. Or in other words, you can say that it contains a lot of chlorophyll to trap light energy. The thylakoid membrane is also where you'll find a lot of electron carriers of the electron transport chain. And these electron transport chain proteins are important to set up a proton gradient. So notice the answers are not out of this world. It's everything you expect to find on the thylakoid membrane. Next, okay, this one is a little bit a leap, but it's also quite relevant. The thylakoid membrane actually is impermeable to ions or protons. Okay, and this is important. If it's very permeable to protons, actually we cannot create a proton gradient. The H+ ions will not store on one side of the membrane, and we cannot set up a proton gradient if it's just fully permeable. Okay, instead, there are proteins that specifically pump in one direction, and the H+ ions will be stuck on the opposite side. It will only be able to flow through again through ATP synthase. And this is really important. Okay, so the idea that the thylakoid membrane is impermeable to protons, except at the ATP synthase and proton pumps where it's necessary. Finally, you could say that the thylakoid sorry, that's a motorbike. Finally, you could say that the thylakoid contains ATP synthase. Why is that important? For the formation of ATP. So, although we are asking something that we didn't quite teach per se, all of these points are very relevant to the thylakoid membrane. It's points that you've seen before. So on the flip side, if I were to put it into context in cellular respiration, I could have a question saying, describe the role of the inner membrane in cellular respiration. And you could describe almost the same things. You could say that, oh, there are electron carriers, set up proton gradient, the inner membrane is impermeable to protons. That way you can maintain the proton gradient. The inner membrane also has ATP synthase. So it's very similar. Let's go to the next question. In the absence of light, the light-independent phase will cease to function. Or in other words, why will the Kelvin cycle cease to function in the absence of light? Because the Kelvin cycle depends on two products produced from the light-dependent stage, ATP and NADPH. Without these two reactants, the Kelvin cycle will no longer be able to function. And we have come to the end of this topical exercise. After you listen to me going through the question, let's do a final review to see if you still feel not confident about various success criteria, or you feel like you've made an improvement in your understanding. In areas that you don't quite understand, please read up on the sections a little bit more. And if you still don't understand, please contact me so that so that all your doubts don't accumulate. Okay, with that we come to the end. Thank you for listening.